Hello everyone, I'm Sheena and welcome to my channel Comprehend Biotech. We are discussing RNA-seq and this is part 3 of the video series. In part 1 and 2, we discussed RNA isolation, RNA integrity number, that is RIN. We learned about RNA species selection and we saw how mRNA is enriched. Then we saw the reverse transcription of mRNA into cDNA. Then we started with the cDNA library preparation, under which we studied how to ligate adapters, what are Y adapters, and after that we studied how to do Bridge PCR or cluster amplification. In part 3 today, we will be learning about what is next generation sequencing and introduction, a basic introduction to it. We'll be seeing what is sequencing by synthesis or Illumina sequencing. Under that, we'll also be studying what is paired end sequencing. So let's start. Welcome again. In RNA-seq, how we design our experiment is very crucial, and that will depend on our end goal, whether we are going to do transcriptome assembly, or are we going to do gene expression studies, or are we going to identify some novel transcripts. So based on that, we will be deciding which RNA species we are going to isolate, how we are going to enrich it, which sequencing platform we are going to use. And based on which sequencing platform we are going to use, we'll decide on how the library will be prepared, what adapters will be ligated to your cDNA, and also which PCR technique will be done to get the necessary amplification. The end goal will also tell us whether we need only single end sequencing or do we need to go for paired end sequencing. So all those things have to be decided beforehand, and that is done during the experimental design. So that is what is being told over here, that when designing an RNA-seq experiment, the selection of a sequencing platform is important and dependent on the experimental goals. Now, currently, there are several NGS platforms that are commercially available. Now, when we first learn about sequencing, we hear about Sanger sequencing and Maxim Gilbert sequencing, right? Out of which Sanger sequencing had become more popular and later on it was automated too. These methods are very accurate, though they take a lot of time and they are very costly, but the end result is the data that you get is very accurate. Later on came the next generation sequencers. Next generation sequencing, or NGS, is a term given to modern high throughput sequencers. NGS uses array-based sequencing, which combines the techniques developed by Sanger in order to process millions of reactions together in parallel. So this results in high speed and high throughput at a reduced cost. But it also comes with a price that the data that you get at the end might be compromising on the accuracy part. Now, majority of high throughput sequencing platforms use a sequencing by synthesis method to sequence tens of millions of sequence clusters in parallel. Now, if you remember, Sanger sequencing is also called chain termination reaction. In case you do not know about Sanger sequencing, please do refer to my earlier video. It was a live lecture that I had recorded on Sanger sequencing, but the concept is well explained. Please do check that out. So there, we refer Sanger sequencing as a chain termination method. As in when a dideoxynucleotide comes and adds, your reaction is terminated. And since that dNTP is labeled, that is how you know which particular dNTP has come and added to your sequence. And that is how we come to know what is the order of sequence. Here, in sequencing by NGS, that is next generation sequences, majority of them use a technique called sequencing by synthesis. And that means here we are going to use a sequencing technique where termination occurs, but it is reversible. So sequencing by synthesis is also called, or Illumina sequencing is also called, reversible terminator sequencing. Okay. Now, out of all the sequencing platforms, Illumina HiSeq is a platform that is very commonly used for RNA-seq. So here we use a sequencing by synthesis method, which we'll be learning in the coming slides. Okay. So as I told, this is also called reversible terminated sequencing, Illumina sequencing, and sequencing by synthesis. So do not be confused. So somewhere you might see an abbreviation like SBS. That is nothing but sequencing by synthesis. So sometimes we refer to Illumina sequencing as SBS technology also. Okay. So Illumina sequencing works by simultaneously identifying DNA bases as each base emits a unique fluorescent signal and adding them to a nucleic acid chain. This is very much similar to what happens in Sanger sequencing. There also you have a fluorescent signal that is being emitted, and that is what you capture. Okay. So where does the difference come? So reversible terminated sequencing differs from the traditional Sanger method in that instead of terminating the primer extension irreversibly using dideoxynucleotide, Modified nucleotides are used in reversible termination. So I'll be showing you what those modified dNTPs are in the later slides. So let's see how the procedure is actually done. Okay. In part 2 of this video series, we had discussed about library preparation, and under that we had seen the last step was amplification. You need to have enough sample to start with. And in Illumina sequencing, we perform something called Bridge PCR. In Bridge PCR, what happens is that you have a flow cell. On that, amplification happens in such a way that in one cluster, you have multiple copies, numerous number of copies of both forward strand and reverse strand of a particular cDNA. This is kind of how the end result of bridge amplification looks like. You can see a cluster over here, you can see a cluster over here, a cluster over here. These represent forward and reverse strands of a particular DNA strand. This might be another DNA strand. These might be different from each other. So we had learned that we can do multiplexing in next generation sequences. All those we had discussed in the second part video. Please do go and check it out if you have any doubt about it. Now suppose this diagram represents one such cluster where you can see both the forward as well as the reverse strand. If you remember, the blue strand was the forward strand, the red strands were the reverse strands. So the first step before you actually start the sequencing, you will be removing all the reverse strands. So you'll be only left with the forward strands of the particular cDNA. Next, 3' ends of the forward strands and the unbound adapters. That means this is a 3' end of each strand, right? And these are the unbound adapters. So here is where your reverse strands were there, right? So they will be chemically blocked. Okay, so you will add certain blocks so that unwanted priming does not occur. Just to refresh, your adapters of P5 and P7 on both the ends look something like this. It has a sequence which helps it to attach to the flow cell. You have an index over here, I5 index. On the other hand, you have I7 index. And next to that, you have the primer binding site. And these primer sites could be either used for reading the DNA insert, or on the reverse strand, they're used to read the index. Okay, so you have a primer binding site. So what happens after your 3' ends and the unbound adapters are blocked is you flood your flow cell with primers. So the primers will come and attach over here. You can see this read one primer site, right? So your primers will come and attach over here. And then the sequencing step will proceed to reading the sequence. Okay, you're going to sequence, or since you're selecting the forward strand, you'll be sequencing in this direction. From 5' to 3' is the new strand formation. So here sequencing by synthesis happens. So while you're sequencing, a new strand will be synthesized in this direction. And as and when the strand is being synthesized, you'll be able to record which base is coming and adding complementary to your initial DNA strand. Okay. So after the primer is added, next you'll be flooding your flow cell with dNTPs and DNA polymerase. Now these dNTPs are special. We are looking for two properties. First thing is we need to be able to identify which particular base has come and added. Second thing is once a base has come and added, we want the reaction to temporarily stop, or else how will the imaging instrument be able to capture an image? Because if continuously DNA polymerase is going on adding a particular base, how will it be able to capture that image? So in order to do that, these dNTPs have two components. Please do listen carefully. This is how your differently modified dNTPs look like. This is your normal dNTP. You can see over here, it has a sugar, it has a phosphate, it has a nitrogen group. On this nitrogenous group, you have a particular fluorophore attached. So for a particular base, for example, adenine, thymine, and cytosine and guanine, each of them will be having a different colored fluorophore attached to them. This is that fluorophore being attached to them. Also, their 3' O, right, it will be blocked by an azidomethyl group. You can see over here N3, right? This is the azidomethyl group. Now, why this is essential? What happens is, once the sequencing starts and the first base, for example, in your DNA fragment, you have the first base as T. We do not know what it is, right? So all the bases will be flown, A, T, C, G. Whichever is complementary, adenine has to come and add. To T, you will have complementary adenine. Adenosine triphosphate will come and add. And since the 3' O is blocked over here, next dNTP will not be able to come and add. So the sequencing reaction is temporarily stopped. Okay, this is a reversible reaction. You can always remove this thing. Okay. Also, whatever color is present in adenine, that will be captured. Okay. So two properties of dNTPs. modified dNTPs that we use in sequencing by synthesis. Please do remember that. So this is the diagram that is going to represent what we are trying to do. Suppose this particular DNA strand over here is representing one cluster. So there will be multiple copies of this particular forward strand, and this, let it be another cluster. To this, we'll be first adding primers. You can see primers are coming and adding over here. Wherever this primer site is, it will come and add over there. Next, you'll be adding dNTPs and DNA polymerase. So in the presence of DNA polymerase, whichever is a complementary base, it will come and add. For example, here you have an adenine. So complementary to it, thymine will come and add. Thymine has a particular color. You can see T is labeled with green color, G with blue, A with a pink, and C with orange. This is just for representation purposes. Okay. And over here, complementary to C, your G will come and add. At this point, what will happen? Another DNA polymerase is already present in your reaction mixture, but another A is not going to come and add over here, or another T is not going to come and add over here, because the chain is temporarily terminated because your 3-O is kind of blocked. You need a 3-OH to form a phosphodiester bond with an incoming dNTP, right? So that bond formation is temporarily cut off. So right now, one particular dNTP has come and added. So that is what is being said over here. A labeled dNTP complemented to the first base of DNA strand ligates itself to the primer. The 3-O of the dNTP is reversibly blocked by azidomethyl group, which prevents any further addition of nucleotides. Okay. So next, what will happen? The unbound nucleotides are washed away. So whatever was unbound is washed away. The flow cell is then imaged. The color captured will indicate which base has been added. So what you do over here, you wash away the unbound nucleotides, and then a camera captures which dye is bound. So for a particular cycle, 4 different images are taken, because you need to take a photo at 4 different wavelengths. Only then you will be able to get the signal from a particular fluorophore. For example, if T is added over here, green color is being shown, right? So only at a particular wavelength, you'll be seeing that green color. In all the other 3 images that you have taken, there will be a blank over there. You'll not see any capturing. So a computer will decipher. It will be able to tell us which particular base has come and added. So in this particular case, you'll be seeing a green dot over here and a blue dot over here. Also, please again remember, this is not the signal from just one particular strand. This is from a cluster. That is how, if there is a cluster only, then that intensity is high enough so that it can be seen or captured. Okay, so this strand is representing a whole cluster. Okay, and this one is representing another cluster. So we have understood that this particular cluster is having a T in the beginning. Okay, a T has come and added. Next, what will happen? The fluorescent branch is cleaved using TCEP, that is tris-2-carboxymethyl-phosphine. TCEP also removes the 3'O azidomethyl group, regenerating the 3-OH, and the cycle can be repeated. So what is going to happen? The fluorophore is going to be cleaved off, and the azidomethyl at the 3' end is also going to be cleaved off. This is going to be done by something called TCEP, that is tris-2-carboxymethyl-phosphine. Okay, so what will happen? You have a 3-OH that has been regenerated, and when the cycle is repeated, next dNTP will be easily able to come and add by forming a phosphodiester bond. So the cycle is continued. So you can see over here, the flow cell is again flooded with modified dNTPs, and the whole process is repeated again. You can see it is again flooded with dNTPs and DNA polymerase. So complementary to T, A will come and add. Complementary to A, thymine will come and add over here. All this will be washed off. Again, the chain is temporarily terminated because 3'O is blocked over here, correct? So this is repeated again. You're going to wash it, you're going to capture the color. So again, you're going to take 4 images, and in whichever wavelength the color has come, the computer will be able to decipher that and do the base calling. That is something called base calling, okay? The computer tells that this is particular base being assigned to this particular fluorophore. So this one has come and added. So that is called base calling. How accurate that is all given by something called a Fred score, which we'll be discussing the part 4 of this video, okay? The cycle is repeated again and again. So after each cycle, you will have one particular nucleotide being added, the reaction being stopped over there, the images being captured, then again the fluorophore being removed, the 3' end being unblocked, and the cycle being continued again and again. Now, if you remember, in library preparation, we had studied that cDNA library might be prepared with fragments which are having 150 to 300 base pair length. Your cDNA fragments might be that long. But out of that, only the ends, that is 50 to 150 base pairs, will be actually sequenced. So that is called the read length. How much is being read? Okay, so that will decide the number of cycles. So if one cycle has been done, one base has been read. Likewise, 50 to 150 base pairs are being read from one end. So that is what is being shown over here. So your fragment length might be 300 base pairs long, but the read length might be, say, from 50 to 150 base pairs. That is how they limit it. And it is being read from one end. Okay. Now you might have a question, why should we limit the read length so much? Why can't we read the whole fragment? Then we can have more data generated. But to explain this, let us just look at one cluster. One cluster will have multiple copies of the same cDNA strand, right? So let us just look at it. What happens is, due to the limitations in sequencing, okay, it is an automated process and it is high throughput, it happens very fast. What happens is, in a particular cluster itself, as the cycles go on passing by, sometimes certain strands might be at a wrong cycle at a particular time. For example, suppose we consider the 100th cycle, and all these strands where you see these green fluorophores being attached, right? These are at the 100th cycle. Suppose this is the 100th nucleotide and this complementary one has come and attached. Just for an example, I'm saying, okay. What happens is, due to limitations in the sequencing procedure, it's a high throughput sequencer, right? And you cannot control everything. There are limitations. And since it is high throughput, there will be chances of errors also. What happens? Some of the strands may be going one cycle further due to some reason, due to some discrepancies in the sequencing method. Suppose this nucleotide, if you look, this is at its 101th cycle. But if you look at this one, this is lagging behind. This might be at the 99th cycle. So when you take the image of this, a lot of it you will see as green color, but there will be mixed signals of yellow and red also coming. So as and when the cycle proceeds, this error will be accumulated, and a stage will reach when you will not have a distinct color that you have captured from the images, because each dot in that flow cell that I showed you, that represents a cluster. So the signal that you're getting, its intensity depends on how well each nucleotide has attached to the fragment. So as and when the cycle is proceeding, this signal will become weaker and weaker. That is why we limit our read length to, say, 50 to 100, even though your fragments might be long, and we do the sequencing from one end. So we are studying sequencing by synthesis developed by Illumina. Now, the particular method that we saw right now, where you had ATCG labeled with different fluorophores, is called a 4-channel SBS, okay, or sequencing by synthesis, because 4 different colors have been used. So in 4-channel SBS, bases are identified using 4 different fluorescent dyes for each base and 4 images per sequencing cycle. So for each cycle, you will have 4 images. That is what I have been telling, right? The sequencing cycle begins with a chemistry step where all 4 differently labeled bases are added to the flow cell. Following nucleotide incorporation, the imaging cycle begins and includes the capture of 4 distinct images using 4 different wavelength bands. This I've just put it in words for you to understand it better. The images are processed with image analysis software to determine which nucleotides were incorporated at each cluster position across the flow cell. Therefore, with 4-channel sequencing, every sequencing cycle requires 4 dyes and 4 images to determine the DNA sequence. Okay. The MySeq and HiSeq series systems currently use 4-channel SBS. So I can explain it better with this particular diagram. So just imagine these are 4 different clusters, okay? Right now we are representing that for this particular cluster, A has been added, for this one G, for this one T, and this one C. These are 4 different clusters or 4 different strands at a particular cycle. Now at a particular cycle, 4 images will be taken for each, right? For each cycle. So these are the 4 images, image 1, 2, 3, 4. So if you concentrate on this particular cluster, you will see in image 1, a color has been seen, in image 2, nothing, image 3, nothing, image 4, nothing. The result is this particular cluster has an A added to it. If you look at this particular one, in the first image, nothing was captured. Second image, it is showing a fluorophore or color intensity is there. Third and fourth, nothing. So the software analyzes and says that cumulatively, it comes to a conclusion that the nucleotide added is G. So on and so forth, you'll be able to find out whether it is T or C. So this is how the 4-channel SBS works, okay? For each cycle, 4 images are taken at 4 different wavelengths, okay? Now, Illumina has also developed two-channel SBS and a single-channel SBS, wherein you use two dyes and a single dye respectively. This is done in order to cut the cost also. Okay. So in two-channel, rather than using a separate dye for each base, two-channel SBS simplifies nucleotide detection by using two fluorescent dyes and two images to capture or to determine all the four base calls. Okay, so images are taken using red and green filter. So there will be a red dye, there will be a green dye, and images are taken in both those filters. Okay, so two images are only taken. So thymines, that is T, are labeled with a green fluorophore. Cytosins are labeled with a red fluorophore. Adenines are labeled with both green and as well as red. Guanines are permanently dark. So whenever guanine is there in both the images, nothing will be captured. That means guanine is added, right? So the mini-seq and the Next-seq and Nova-seq systems are the ones that use the two-channel chemistry. So this is an example that shows two-channel chemistry. So you look at this particular cluster, these are four different clusters, two images are taken. So if there was an A added, since it has both green fluorophore as well as red fluorophore, when you look at image 1 and 2, both of them, the color will be there. So the computer will be able to say that it is an A. If it is G, since there is no fluorophore, it will be always dark in both the images. For T, it will be having image only in the green filter or green wavelength, whatever wavelength you're using to detect the green fluorophore. Same way for C, it will be only in the red one. So that is how you detect AGTC in a two-channel chemistry. The latest development by Illumina is one-channel SBS. So let us see what happens there. So the iSeq 100 system uses one dye, two chemistry steps, and two imaging steps per sequencing cycle. So the dye number is reduced, but you still have to do two imaging steps. Also, there's in between a chemistry step. Okay, we'll be seeing what that is. So in one channel chemistry, adenine has a removable label and is labeled in the first image only. Cytosine has a linker group that can bind a label and is labeled in the second image only. Thymine has a permanent fluorescent label and is therefore labeled in both images, and guanine is permanently dark. Okay. Nucleotides are identified by analysis of the different emission patterns for each base across the two images. So this system uses a pattern flow cell with nanowells fabricated over a complementary metal oxide semiconductor, CMOS chip. Okay. So I'll just explain to you what happens over here. In this, adenine is there, no? It has a removable label. Label is nothing but your fluorophore. Okay, one single dye is used. This dye is attached to adenine in such a way that it can be removed. Okay, guanine has nothing, no fluorophore, same as before. Thymine has a fluorophore which is permanently attached to it. It cannot be removed. And cytosine has a linker over here. This linker helps in attaching the cytosine with this particular fluorophore in the later stages, while the second image is taken. Before the first image is taken, you will add dNTPs in such a way that A has a fluorophore, G has nothing, T has a fluorophore, labeled fluorophore, and C has only a linker adapter. There's no fluorophore at that point. Okay, and so you will flood these dNTPs over your flow cell. So what will happen when you take the first image? If A is there, there will be a fluorophore. G, if it is attached, there will be nothing. It is permanently dark. T will be obviously shown. And in the C, since there is only this linker over here, it will also not show any image. After this first step, now they do a chemistry step. What they do in that step is they remove the fluorophore from the adenine or the label from adenine, and a label is added to the linker present on the cytosine. After this step, what will happen in the next image, since there's no fluorophore in A, nothing will be seen in A. And in G, obviously, there is nothing, so it will be dark. T is permanently having a fluorophore, so it will be having a color. Whereas, since newly a fluorophore has been attached to C on this linker, it will be the image will capture a fluorophore or a fluorescence over here in C. So in the final step, software will analyze, and looking at both the images, it will be saying, since it is fluorophore over here and nothing over here, this is A. Both dark, this is G. Both fluorescence signal is there, that means this is T. And only in the image 2, if it is there, this is C. So that is one channel SBS. So that has also been developed. This is a very recent development. Okay. So this is Illumina sequencing, or sequencing by synthesis, or reversible terminator sequencing. Okay. So after doing the sequencing, right, the data that you get will be in the form of a graph. And analyzing that, a software will already have done the base calling. Also, with that, you will have the Fred scores or the quality of the base calls. So that we'll be discussing, as I told, we'll be discussing the next part. That is what we get. Okay. Now, depending on our experiment goal, we will be deciding whether we need to go for single end sequencing or paired end sequencing. So that will depend on what we need at the end. Do we need to do transcriptome assembly, or do we need to do some gene expression studies? Based on that, we'll be deciding whether we want single end or paired end sequencing. Before that, let us see what is single end sequencing and what is paired end sequencing. Okay, suppose this is your cDNA fragment, which you have got after library preparation step. Okay, you're going to send it for sequencing. If you read it from one end, that is called single end sequencing. Okay, you're going to choose one end and you're going to read it. Mostly it is from that, if your forward strand is there, it will start reading from the 3' end so that your new strand is synthesized from 5' to 3', right? So what is paired end sequencing? You will have your DNA strand and you will have the reads from both the ends. That is from this end as well as this end. Okay, and how this is done? It's not as simple as it looks that you start reading from here and then from here. No, you have to do an in-between step in the sequencing part. We'll be seeing that. Before that, what would be the need of going for paired end sequencing? Let us look at that. Okay. Now, the use of paired end sequencing is not limited to RNA-seq. I'm just discussing it under RNA-seq because we have just learned how sequencing is done. So it will be useful if you know about paired end sequencing. Okay. Now, suppose you want to carry out the assembly or sequence alignment of a newly sequenced DNA fragment. So mostly what we do is if we have an unknown fragment of DNA, we try to map it using a reference genome. Okay. So with that, we'll try to understand the importance of paired end sequencing. So suppose this is your reference genome, and these are the fragments that you have got from new fragments, okay, cDNA fragments or any DNA fragments that you have from the new DNA strand, which has still not been mapped, okay. So you will try to align these fragments to your particular reference genome. I have colored these particular areas just to differentiate between the different regions, okay, so that we understand where the overlapping is happening. Now, suppose you're doing something called single end sequencing. Single end sequencing means you'll be reading it from one particular end, okay? And suppose this is your read length. Read length means how much of it is being read in particular run of a sequencer, okay? So as I told you, your cDNA or DNA fragments might be long, but your reads might be of a specific length from 50 to 100 base pairs, suppose, okay? So what you will do, you will carry out the sequencing. All these fragments will be read at their particular ends. For example, you can see over here, right? We are going to read all these ends from a particular end. Now, suppose this particular reference genome had a repetitive area. You can see over here, this is a repetitive area, this is a repetitive area. I've drawn it over here, okay? After sequencing, what you get over here, this particular fragment, I have written the corresponding nucleotides over here to give a representation, okay? So for example, this is starting from TGGC and then you have a green fragment over here. Of course, these colors are not seen and we have no clue, okay? We just have the basis. Same way over here, this is starting with TGGC, okay? Now, where will the problem arise? The read will be of, say, this particular length. And please know, this is just 5 nucleotides I've shown over here, but it will be way too long. Read length will not be 5. It will be between 50 to 100, okay? It's just for representation. Suppose your read length is giving you from one end only so much. What could be the confusion that you might have? This read is only so much, TGG, suppose. The confusion will be whether this TGG belongs over here or does it belong over here, because it is a repetitive region. You will not know whether to place it over here or over here because you have no clue what this part is. That is where paired end sequencing comes in play, because in paired end sequencing, you will read it from this particular direction. Okay, so when you read it from this particular direction, you will have a read which represents this area. You will also have a read which represents this area in each of the fragments. So once you have this and this corresponding to a single fragment, you will be able to place this particular read over here. You will be able to note that this is particularly green area. Green means these particular nucleotides corresponding to this one. And this is, since these are not repetitive, right? You'll be able to understand this is the read over here. Okay, so you'll be easily able to place this fragment over here and this fragment over here. There'll be no confusion. I hope that is clear. Why would we need paired end sequencing, right? So paired end sequencing actually aids in sequence alignment a lot. Okay. Now, before we jump into how paired end sequencing is done, you need to look at the adapters once again. So this is the P5 end, this is the P7 end. We said that this is the area which attaches to the flow cell. This is your index area. This is your primer attachment site. This is your DNA insert. Okay, and here you have the primer site for I7 index. Here you see I7 index. Then this is your P7 adapter, which attaches to the P7 adapter on the flow cell. Okay, same way for the complementary strand also. Just keep this in mind. Let us go into what happens in paired end sequencing. So in single end sequencing, we saw, right, the first step was you remove the reverse strands and you're only left with forward strands on the flow cell, and the read is taken for the forward strand, right? So that is what is being shown over here. This is your forward strand, suppose. These are the different areas. This is your I5 index, and this is your primer site for the read one primer. So what will happen is once you start sequencing, in single end sequencing, what happens is your read one primer comes and attaches over here. That is what we saw in the sequencing also, that primer comes and attaches, right? So this is the site where it will come and attach. Your sequencing will start taking place. So the second strand will be synthesized over here, and as and when each nucleotide is added, image is captured and you get the read, right? Now this I have shown over here till here, the second strand being synthesized, but no, not necessarily that. Now, if this is some 300 base pairs long, only till here the read might be taken. Okay, this is just for representation. I have shown that the second strand is synthesized, and while it is being synthesized, it is read. That's why it's called the read one. Okay. Next. In the next step, what happens is this particular strand is dissociated. There are different chemical and thermal conditions that are changed in order to cause these dissociations. Okay, so read one is dissociated. Next, what will happen? This is the primer site for I7 index. So if you want to read I7 index, here is where its primer will come and attach. So in the next step, I7 index primer will come and attach over here. And again, when the sequencing is going on, this particular I7 index will be read. Okay, so I7 is called, index is called the index 1. Okay, which corresponds to the read one. Why do we need indices? In order to identify that this particular fragment is from this particular sample, especially when we are doing multiplexing, right? When we take samples from various experiments, we need to give each one a particular ID or a particular tag. Again, in case if you do not understand what is multiplexing, please refer to the part 2 of this video where we have discussed about that. Okay, the importance of using indices and barcodes. Okay, so along with read one, I7 index will be also read. So both of these together will be associated with each other. So we will know I7 index, we will know the read one. Okay. Next, what will happen? This I7 index is now dissociated. Okay, this I7 index is now dissociated. So till here is the single end sequencing. You have sequenced a particular read. You also have its ID tag. Okay, both of them are done. This is single end sequencing. Now, if you want to do paired end sequencing, you're going to have to sequence this particular strand from this end. How do we do that? For that, we will be first removing the 3' blocks that were there, and also the unbound adapters were there, right? They were blocked, right? We had discussed it in the beginning step that after reverse strands are removed, this 3' end of the forward strand and the unbound adapters on the flow cell are actually blocked. So after your first read has been taken and the first index has been sequenced, you will remove this 3' block as well as the block on the adapter. Those both are removed. Next, what will happen? This particular adapter will go and find its complementary adapter on the flow cell, bend down, and bridge is formed, right? If you remember Bridge PCR, you will know how this is done. Please refer back in case you have not, if you're not aware of it. In part 2 video, we have discussed Bridge PCR, okay? So this particular strand will bend down and a new strand will be synthesized like this, the complementary strand. And as you can see, this was your blue color was your forward strand. Complementary to it, a pink, red strand has been synthesized, okay? So that is a newly formed strand. After this, what will happen? You're going to dissociate this original forward strand. This forward strand will be removed now. Okay. So this one is already red, right? It is red from this direction and the index is also red. This is going to be removed. You have its complementary strand over here because once these two strands dissociate now, this will stand like this and this will be standing like this. This particular strand will be removed, forward strand will be removed. You will be left with the complementary reverse strand. Now the same thing will be repeated. Only difference is this particular area, which was actually acting as a read 1 primer site over here, here it will act as a I5 index primer site. So I5 index primer will come and add over here, and this particular index, I5 index, will be sequenced. After I5 index is sequenced, that will be dissociated, and this particular area, which previously in forward strand acted as the I7 index primer site, will now act as a read 2 primer site, and the read 2 primer come and attach, read 2 will be taken. Okay, so it is taken from this end. So now you will have read 2 primer and I5 index. I5 index is called index 2 because it is corresponding to the read 2. It will identify the second read. Okay. So in this way, a particular strand has been read from both directions. Now, an obvious question is, this is a complementary strand. The other one was a forward strand. One is a forward, one is reverse. Then how are you saying that a same strand is being read from both ends? The answer is, your softwares already know that you're reading the complementary strand. So they take the reverse complement, and that is how your data is given to you. So when you get the data after read 2, it will not be the raw data that you sequence over here. This read 2 will be reverse complemented, and then that data is given to us. Okay, so I'll explain that with an example. So with this diagram, I'll just explain to you how that reverse complement thing will actually give us the ends of the same fragment. Okay, suppose this was your forward strand. It is forming a bridge and form second strand, that is a complementary strand. So after that has been done, you have the forward strand as well as the reverse strand. Okay, this will be read from this area, that will be read 1. This will be read from this area, that will be the read 2. Okay, what do we do with it? So suppose this is your actual forward strand. This is the same thing I have represented over here. Okay, so what will read 1 give me? This is just an example. I have just taken 4 nucleotides as read 1. Of course, it will be longer. Okay, just to represent. Read 1 will be 3' GCTT, right? It will read from here. So it will be 3' GCTT. So what will read 2 give us? Read 2 will be read from 3' end of this reverse strand. So it will be 3' ATGC. So that is what is being shown over here, 3' ATGC. But this is not how we get it. Softwares will do the reverse complements. So let us see how that will give us the end result. Okay, so if 3' ATGC, we take the complement, what will be the complement of this? It will be 5' TACG. So that is what is being written over here, 5' TACG. Then this will be reversed. The reverse will give us GCAT 5'. Okay, GCAT 5'. So you take the reverse complement. So by the end of this reverse complement, what are we getting? If you look at read 1, it is 3' GCTT. If you look at read 2, it is 5' TACG from this end, right? So this is corresponding to this, and this is corresponding to this. So you have read a particular DNA fragment from both the ends. Okay, so this is how paired end sequencing is done. Even though you're reading it on the complementary strand, softwares are doing the conversion and giving you the final read. Okay, I hope that is clear. With that, we come to the end of part 3 of this video series. We saw what is NGS, we saw what is sequencing by synthesis by Illumina, and we saw what is the importance of paired end sequencing and how it is done. I hope you understood the concept. In case you found this useful, please do let me know in the comment section. Please do like this video, and please do not forget to subscribe to my channel. It will be a great help for me. I will meet you with the part 4 of this video where we will be looking at data analysis part. And until then, bye-bye.